Thanks for coming to the lecture today. I'm Kate, I'm one of the 5th year medical students, and I'm going to be talking about nerves and the neuromuscular junction. And this is a lecture for first year Phys and Pharm. I really liked this topic when I did it, so I've tried to include some bits from my essays and my tutorial feedback that I had in first year. This is your spec, so I've tried to include everything on there. I've labelled the slides with a green dot on the top right, which means most of the material on there is core, and then a red dot if it's extra on the right hand side of the spec, or experimental evidence or something like that. So hopefully that's useful. So starting by going back to the basics, because there are some terms that we're going to use throughout the whole lecture, which are important to get your head around. So charge, ionic charge, a positive or negative charge carried by an atom or a compound, important because we talk about lots of different ions on this. Current is the rate of flow of charge, not to be confused with voltage, which is how difficult it is to make charge flow. So the energy required to move one unit of charge from one place to another. And potential difference in voltage are going to be used interchangeably here. Resistance is the voltage over the current. So the equation is V equals IR. And that's the potential difference required to attain given a flow of charge. Conductance, which is the inverse of the resistance, so just 1 over the resistance, and that means it's how easily charge will flow. And capacitance is the ability of a circuit to store charge. So kind of you can think about it like a battery. The first thing we're going to cover is the resting membrane potential, and then we'll talk about action potentials and then the neuromuscular junction. So the resting membrane potential is the voltage across a plasma membrane at rest. And the value of the resting membrane potential is generated by an unequal distribution of ions on the inside and the outside of a cell. And that relies on and is affected by the ionic concentration gradients and the relative permeability of the membrane. Resting membrane potential controls the osmotic balance of cells and regulation of cellular volume via maintenance of correct ion concentrations. And this control stops cell swelling and prevents lysis, so it's really important. This is a diagram, and this is similar to a diagram that I drew in my first year essays, showing there's a higher concentration of potassium in the inside of the cell. Sorry, this is a cell when this is your phospholipid bilayer. So inside the cell, there's a higher concentration of potassium, and there's a higher concentration of negatively charged anions. And these are amino acids and proteins which are produced by the cell and normally have a constitutional negative charge. And then on the outside of the cell, there's a higher concentration of sodium, chloride, and calcium, and some other ions which I haven't included. So in every cell that we're going to be studying, and in here we're mostly looking at neurons, there is a sodium potassium ATPase, which drives the exchange of 2 potassium into the cell for 3 sodium out of the cell. And this establishes a concentration gradient for both potassium and sodium within the cell. And the sodium concentration gradient is inwards, so the sodium that's been pumped out is wanting to move back down its concentration in, and potassium's concentration gradient is outwards. And that's really important here. Talking about the potassium concentration gradient, due to the sodium potassium ATPase, the potassium concentration on the inside of the cell is normally about 145 millimoles per liter and 5 millimoles on the outside. So there's a strong concentration of this potassium wanting to move out of the cell. It can't diffuse through the phospholipid bilayer. None of the ions can diffuse through because of their charge, but potassium can move through potassium leak channels and potassium inward rectifier channels. And as it moves through these channels, it leaves behind these negatively charged anions, which it was associated with, and leaving these inside the cell. And as more anions build up inside the cell without potassium to equalize that charge, there's a negative charge that builds up in the cell. And in turn, this attracts some potassium ions back into the cell. And that's called the electrostatic gradient. So we have the potassium concentration gradient going outwards, and then the potassium electrostatic gradient going inwards. And the electrostatic gradient doesn't actually require many potassium ions to move back in, so it doesn't actually affect this concentration gradient that's already established. It only requires a couple to equalize that electrostatic gradient. So this is just kind of what I've been talking about, but at the point where the concentration gradient is equivalent to the electrostatic gradient. So when equilibrium has been reached, this is called the equilibrium potential or the Nernst potential for potassium. And for potassium, this is about -92 millivolts. And some of you who know your resting membrane potential will know that that's not the same value as resting membrane potential. Resting membrane potential between -70 and -75 millivolts. But the reason for this is obviously potassium is not the only ion that affects the resting membrane potential. So you have to work out the equilibrium potential for all the other ions that are also crossing that cellular membrane. And when you calculate those together, that's the resting membrane potential for the entire cell. So this slide just summarizes kind of what we've just spoken about, and shows the different types of channels that are in the membrane. So you have your ATPase, and you have sodium channels which are closed at rest, which is why the sodium doesn't move, and the potassium is the one that's able to leak through at rest. The way we calculate the equilibrium potential, so what I just spoke about, the Nernst equation for potassium, to get that numerical -96 value, we have to use the Nernst equation. So I'm talking through the scary looking maths in the middle there. I promise it's not that scary. So E, which is the membrane potential, is equivalent to 61.5 divided by Z. Z here is the charge on the ion. So for potassium, it's 1. For sodium, it's 1, whereas for calcium, it would be 2. Multiplied by the log of the concentration of the ion inside the cell, divided by the concentration of the ion outside the cell. So the actual resting membrane potential is somewhere between all the different resting membrane potentials for the different ions. And potassium here actually has the biggest effect because of that steep concentration gradient that we've been talking about. So this is the Nernst equation. And this allows you to calculate the membrane potential for individual ions. And we'll get onto how you can calculate it for the entire cell, because it's a little bit complicated when you combine them. So this is like our first piece of experimental evidence. So there was an experiment done in 1959 by Hodgkin and Horowitz, and they used frog muscle cells, and they changed the extracellular potassium that was bathing the cell, and then they measured the resting membrane potential of the cell as they changed this extracellular potassium. And this is the graph. Sorry, it's not very good quality. This is the graph on the right-hand side, which shows the values that they found. And you obviously can see for most of it, it's quite a linear relationship. And it shows close reliance on extracellular potassium concentration. So that's a really important determinant in the changing resting membrane potential of the cell. And there's actually deviation if you look at the graph on low values of extracellular potassium. And this is because there are other ions involved which have more importance at lower potassium concentrations. And so we'll get into that. But this is quite a good piece of experimental evidence that shows how important that concentration gradient of potassium is to establish the resting membrane potential for the cell. This is a slightly more complicated equation, and this is because this helps us calculate resting membrane potential with all the different ions that are involved. So when a membrane is permeable to two different ions, the Nernst equation can no longer be really used to get a precise value for the membrane potential. So this is the Goldman-Hodgkin-Katz equation, and this is used as it's applicable to a membrane permeable to both potassium, sodium, and chloride ions. This equation itself also has some limitations as it doesn't account for the sodium potassium ATPase pump, but that only has an effect of about -4 millivolts. So the reason we're talking about chloride here as well, just an aside, it's the third most important ion in resting membrane potential. Chloride is not actively pumped, but it can move in and out of the cell via chloride leak channels, and therefore it helps equilibrate at a reversal potential in accordance to the potential gradient that's across the cell membrane. And this is normally very close to the resting membrane potential, which is determined by other ions, namely potassium. So our equation here, you can see, again, it looks complicated, but it explains that the resting membrane potential is our constant again. So you remember before that was minus 61.5 times by a logarithm of the concentrations and the permeability of the different ions. And this is extra. You don't need to be able to memorize or do any calculations with this equation, but it's quite important to show how we actually go about calculating the resting membrane potential. And it shows us that the greater permeability and concentration gradient of an ion, so for example, potassium with a high concentration gradient and a good permeability, the greater its impact on resting membrane potential. Then at the bottom, I've just included a table of some of the different resting membrane potentials of different types of cells. So most of the time here, we're going to be talking about neurons. So that's why I've said -70, similar to -65. But other cells have different resting membrane potentials, and that's established by different ions and different permeability of the different membranes. Okay, this is an interesting effect that happens across a membrane. So negatively charged proteins, when they're inside the cell, are unable to pass through that membrane. So this creates an uneven electrical charge, and that's the Gibbs-Donan effect of an unequal distribution of permanent charged ions that can't move. So that's a single Donan effect. But also the low permeability of the membrane to sodium ions means that there's also a buildup of sodium ions on the outside of the cell, as well as the fact that the sodium potassium ATPase pumps sodium ions out. So that's a second Donan effect. That's the double Donan effect. And this is really important in understanding those two concentration gradients across the membrane. So impermeable anions inside the cell and semi-impermeable cation on the outside of the cell. And this helps with cell volume homeostasis, so important in preventing cellular lysis. And this is a diagram just to help explain the concentration gradients of those two things. A little bit of clinical for you. Again, this is extra, but nice to include in an essay. And so we've just spoken about how important potassium is. So you can imagine when you're varying potassium, which is what hypo and hyperkalemia mean, low and high potassium, you get cellular effects and you get effects on the body. So in hypokalemia, and this is defined as when potassium is less than 3.5 millimoles per liter. This is extracellular. It causes hyperpolarization of the resting membrane potential. And this often can present with fatigue, leg cramps, weakness, constipation. So you can see on the left-hand side, this is how it affects an ECG. This is more for interest. You'll cover this in clinical school. And you can correct it by giving someone potassium back so that the concentration comes back up. And then on the other side of the spectrum, you can have hyperkalemia, which is when the concentration is above 5.2, and you get depolarization of the resting membrane potential, because what we've just been chatting about, the balance of sodium, potassium, and chloride is disrupted when the extracellular potassium is changed. And this presents with muscle weakness, paralysis, palpitations, nausea, and vomiting. And you can correct this with a drug called calcium gluconate, which helps reduce the extracellular potassium back to normal. Okay, so now we've talked about resting membrane potential, we're going to move on and talk about action potentials. And it's important to understand resting membrane potential because that's what changes when an action potential is sent down a neuron. So what is an action potential? It's a rapid changes in resting membrane potential that transmit impulses. And neuronal action potentials are important for all nervous communication, including pain and muscle contraction. And we'll talk about some of the clinical side effects of when action potentials don't fire properly. So this is a diagram. I'm sure you've seen something like this before. And this shows the passage of an action potential over time. So you have a stimulus at the start and you have a depolarization where voltage-gated sodium channels open and you have sodium influx into the neuron. Remember, that's down the sodium concentration gradient because sodium is being pumped out, so it wants to move in. And you have this big increase in resting membrane potential as all of these positive ions flood into the cell. And then you have a refractory period. There's the two sections on the bottom. The ARP means the absolute refractory period, and the RRP is the relative refractory period. So the the absolute refractory period is when another action potential can't be triggered because these voltage-gated sodium channels have opened and then they've become inactivated. So even if you were to send another signal down the neuron, they couldn't open again and have more sodium influx. And then the relative refractive period was a period where another action potential can be triggered, but it would require a greater stimulus to bring the resting membrane potential up to threshold. And that's because you can see in the graph that that's fallen underneath our resting membrane potential. So it's underneath our -70. So it would require more of a stimulus to bring it above that threshold. And the threshold here on this diagram is at -55, but it ranges upon which neuron and which tissue you're in. And this is some facts about the action potential. So it's an all or nothing event, and that means that as soon as sufficient voltage gated sodium channels have opened and there's enough sodium influx into the neuron, loads of them open and it causes a huge spike. So you can't have kind of a half action potential. It's an all or nothing. It either goes or it doesn't go. And then we talked about these periods, and then there's propagation of the action potential down the neuron via local currents, and we'll talk about that. I mentioned the sodium channel becoming inactivated. This is an important mechanism to understand. This is extra, but I think it helps understand the action potential. So these are the sodium channels, and they're voltage gated sodium channels, and then they're closed at resting membrane potential. And depolarization causes opening of a voltage gate, and this is called the M gate, which is not labeled on the diagram. And then there's a positive feedback loop, so many other voltage gated sodium channels open. And then they are self-inactivated. So this is the third state on the diagram, by closure of the H gate, which is like the little ball on a string there, which you see inactivating the channel. And this is important because it allows that action potential to repolarize. There's some experimental evidence behind showing that H-gate works. Some scientists use H-gate pronase, which is an enzyme, which basically inactivates the inactivator. So it stops the H-gate from working, and showed there was no repolarization of the membrane, and action potentials couldn't be conducted along the neuron because the sodium could still move through the sodium channels, and if they weren't inactivated. Okay. A bit of extra again, but this is some experimental methodology here. And there are two types of clamping that are used to study membrane potential. There's voltage clamping, where you have a micropipette electrode inserted into a neuron. A current is injected to hold the resting membrane potential constant, and then you get a readout. Or patch clamping, where you isolate a single channel, so you can really use this to study single sodium or potassium channels. And that's really useful as well, just because we're going to talk about patch clamping. So it's good to know the difference between these two. This is some more experimental evidence. Hodgkin and Huxley did, this is a very famous experiment they did on a giant squid axon. And they were basically showing that the action potential was a large, quick, self-inactivating influx of sodium, and that was the cause of an action potential. And you can see that the diagrams on the right-hand side show what action potentials looked like when they used different blockers. So they first showed that it didn't occur in sodium-free solution. So they put a neuron in sodium-free solution and tried to stimulate an action potential, and it didn't work because there wasn't any sodium to flood in. And then they used TTX and TEA. So TTX blocks voltage-gated sodium channels, and hence no action potential occurred at all. And TEA blocks voltage-gated potassium channels, and so no repolarization occurred. So you can see that in the second one is TEA, and the third diagram is TTX there. And this was really important because it showed that sodium was the mainstay of the mechanism of the action potential. This is a slide talking about the fact there's a large difference in conduction velocity between different types of nerve fibers, and we're going to go on to explain how that's achieved. So these are the types of nerve fibers that you see in your body, and they carry different types of signals. So you can see type 1 carries proprioceptive information, whereas type 2 carries mechanoreceptive information. And this is important because it allows signals to be propagated at different speeds. So it allows us to have different timed responses from a single input stimulus. So if you were to touch a hot surface you're actually aware that you're touching it before you're aware of the pain, and that's because pain is carried in a slower fiber than fine touch, and so it allows you to respond in a complex way to a single stimulus. So propagation of action potentials along a neuron is reliant on these local currents that I mentioned earlier, and it's also reliant on saltatory conduction, which is when the conduction jumps between areas of non-myelinated parts of the neuron. So the local currents are formed as the sodium floods in, it diffuses along the axon, and causes depolarization of adjacent sections of the membrane. So that's simple conduction, that's simple local current. And then the area behind the action potential, so if the action potential is moving, the area behind the action potential ensures that conduction only happens in one direction because the voltage-gated sodium channels are still in their inactivated refractory period. However, you can see in the diagram here, there's sections that are myelinated, and this is by Schwann cells, which are quite fatty cells. They provide a high electrical resistance around the neuron. And myelination allows for faster transmission of nerve signals because they can jump between the nodes of Ranvier. Sorry, the nodes of Ranvier are the bits that are unmyelinated in this diagram. And this is called saltatory conduction. And so this allows action potentials to travel really quickly down neurons, and those that are not myelinated have slower conduction. There are two things that determine conduction. So conduction is subjective to both length and time constants that determine the conduction velocity, dependent on a number of different factors. And these two factors can be used to understand how things like myelination and diameter affect conduction velocity. So this is the length constant, which is the square root of the membrane resistance over the internal resistance. So this tells us that if you increase the diameter of a nerve, by increasing the diameter, you decrease the internal resistance, and hence you have an increased length constant, and hence increased conduction velocity. And myelination, for example, increases the membrane resistance because you've got this fatty Schwann cell on the outside of the neuron. So increase the membrane resistance and hence increases the length constant and increases the conduction velocity. The other constant that we talk about is the time constant. So this is the time taken for the nerve to reach 63% of its final change in potential. And it is calculated by the membrane resistance times by the membrane capacitance. And if you have a smaller time constant, it means there's a faster conduction. So for example, again, myelination decreases the membrane capacitance and hence decreases the time constant and hence provides faster conduction. And this is just a summary of what I've just spoken about, just more useful for your notes, but explains myelination, the effect of myelination on conduction. And for example, the effect of temperature. If you have a higher temperature, increased ionic kinetic energy, increased diffusion, and so ions move quicker across the membrane, so you have higher conduction. This is clinical. Again, useful for your essays, but it's extra to the core. So calcium affects action potentials, and the way it affects action potentials is the calcium ion transiently blocks sodium channels, and it shortens the time course for closing of the activation gates. So you get a difference in action potential when you have either low calcium or high calcium. So in low calcium, remember we said calcium ions block sodium channels, so you have reduced sodium channel blocking, increased spontaneous nerve firing, and you have increased tetany. And tetany is when action potentials combined to provide a continuous signal. So this is dangerous in the body because you can have muscle firing all the time, and this can lead to bronchospasm, type 2 respiratory failure, and it's really dangerous. And we help patients who have this by giving calcium back to increase that calcium. And on the other hand, hypercalcemia, so high calcium, you have more channel blocking. And so you get the opposite. So you get muscle weakness because the muscles can't work as well. And it's less dangerous, but you can manage it more conservatively with fluids, because often patients are dehydrated, so their calcium is just relatively increased. So if you give fluids, it dilutes it. Okay, a bit more clinical. This is about types of demyelinating disease. So the reason we're talking about demyelinating disease is myelin helps conduction. So if you have a reduction in myelin, you're going to have less good conduction. So one of the most important diseases which is affected by myelination, is multiple sclerosis. And in multiple sclerosis, there are autoantibodies against the myelin proteins. So you have demyelination, neuroinflammation, and nerve damage. The symptoms can be very variable, but you can imagine that if you have reduced myelin, reduced conduction, reduced ability of your nerves to work, you can get a whole range of symptoms, some of which are listed here. And on the other side, in a more short course, is Guillain-Barré syndrome, or Guillain-Barré syndrome, where you have T-cell attack and autoantibodies against peripheral myelin proteins. So this happens in the peripheral nervous system, and this causes a flaccid paralysis, which sometimes requires ventilation because it can affect the muscles in your diaphragm. And this can be treated with plasmapheresis, which is filtration of the autoantibodies out of your plasma, out of your blood, or IVIG. Again, this is extra, but quite nice to throw a couple of examples into your essays. And the last section of the talk today, so we've chatted about the resting membrane potential, the action potential, and now we're going to talk about the neuromuscular junction. So the neuromuscular junction, this is a diagram. And the neuromuscular junction is a junction between an alpha motor neuron here and the muscles of a skeletal, and the muscle fibers of skeletal muscle, which form the motor end plate, which is what you see in pink at the bottom here. It's chemical transmission through neurotransmitter release, and it allows contraction of the skeletal muscle and movement of the axial skeleton. I've just added this in because this is actually the diagram that I drew in my essay about the neuromuscular junction in first year. So just kind of an example of what you can draw in an examination setting. And I was always told by my tutors to use different color pens and make sure it's really obvious what you've labeled and put numbers on, which I actually haven't done on this diagram, to help explain what you're talking through. So we're going to talk through different parts of this diagram. So acetylcholine, ACH, is the neurotransmitter, and it's released via exocytosis of synaptic vesicles at the postsynaptic terminal. An incoming action potential causes depolarization of the membrane of the end of the neuron here, and it opens voltage-gated calcium channels. And this allows calcium influx into the end of the neuron. Calcium, I'm going to mention some proteins here. Some of these are extra, but some of them have clinical relevance, so it's important. Calcium binds to synaptotagmin in the synaptic terminal, and synaxin, which cause interlocking of the SNAP25 and synaptobrevin, and these form the snare complex. You can see this labeled in the left-hand diagram, and this moves the vesicle close to the presynaptic membrane for release. And the clinical relevance here is that Botox, botulinum toxin, cleaves the snare complex, specifically SNAP25, irreversibly, so the NMJ transmission is permanently stopped. So if you have Botox, you can't recover the same functionality of exocytosis of the vesicles. So acetylcholine leaves via exocytosis and it diffuses across the cleft. And if it's not hydrolyzed by acetylcholine esterase, which is an enzyme that sits in the cleft, it will bind to the nicotinic acetylcholine receptor on the sarcolemma, on the muscle fiber. Two acetylcholine molecules bind to the alpha subunits on the nicotinic acetylcholine receptor, and it causes a conformational change where it opens a non-selective cation channel, which allows both sodium and potassium. Allows sodium in and potassium out, which causes depolarization of the muscle sarcolemma. And you can see in the diagram on the right here, these receptors are bound by MuSK, which is muscle-specific kinase, and we'll come back to that later because it has some clinical relevance. And it was shown that it was calcium that caused the exocytosis of ACH, of acetylcholine, as there was no release when the terminal was in a calcium-free solution or when voltage-gated calcium channels were blocked by verapamil. So quite a lot of information there, quite a lot to get by, but it's a good process to get your head around because it need to understand it to talk about some of the clinical relevance later. So this slide summarizes the life cycle of acetylcholine. So it's synthesized from choline and acetate, acetyl-CoA, by choline acetyltransferase. It's stored in vesicles. It's released, and then we talked about released into the cleft and binds to synaptotagmin. Two of these ACH molecules bind the alpha subunits of the receptors, and it's hydrolyzed by acetylcholinesterase at the muscle membrane back into acetate and choline. Acetate diffuses back into the terminal, and choline is transported in via a sodium choline co-transporter, and ACH is reformed in the terminal by acetyltransferase. Okay, and this is the nicotinic acetylcholine receptor. It's quite a complicated receptor, and you can see in the diagram 2 slides ago, there are 5 subunits, 2 alpha, 1 beta, and 1 gamma and 1 delta subunit in muscle. And it's actually, it varies if you look at different nicotinic acetylcholine receptors in the body. And the acetylcholine molecule binds at the interface between the alpha and the delta epsilon subunits. This is how some of our toxins affect the neuromuscular junction transmission. So transmission at the NMJ can be interfered with pharmacologically by neuromuscular blockers. You need to inhibit at least 70% of nicotinic acetylcholine receptors to stop successful transmission. And that's because there's excess sodium and there's excess acetylcholine. So you have to stop almost all of them to stop transmission. Non-depolarizing, so I'm talking about the stuff on the left now on the core side. So non-depolarizing blockers bind to the nicotinic acetylcholine receptor and they don't open the channel, but they increase the competition for acetylcholine binding. And this decreases the amount of nicotinic acetylcholine to nicotinic acetylcholine receptor acetylcholine complexes, and hence it decreases transmission. And this can be overcome by giving anticholinesterase inhibitors like neostigmine. Depolarizing blockers like suxamethonium are agonists to the nicotinic receptor, and they open them and cause paradoxical depolarization. There's initial muscle contraction and then there's relaxation. So this blocker will eventually get broken down and transmission begin again. It's actually broken down by something called pseudocholinesterase. So suxamethonium broken down by pseudocholinesterase. And there are some patients who actually have pseudocholinesterase deficiency, which we don't know often before they have an operation. And so they can have prolonged apnea after giving a depolarizing blocker, and it can only be broken down in the liver. So patients who have this and who have had anesthetics, for example, need mechanical ventilation until they wake up again. And I talked about the acetylcholinesterase inhibitors like neostigmine. On the right-hand side here, this is not core, but this is just some extras. So black widow venom forms a calcium permeable pore in the presynaptic membrane, so that's a neurotoxic. And the cone snail produces a toxin that inhibits N-type voltage-gated calcium channels, which are involved in this transmission of this calcium influx. This is an experiment which took me a little bit of time to get my head around, but it's important to understand quantile release at acetylcholine at the neuromuscular junction. So Fat and Cats here, which is a good set of names to remember for your exams, they measured the end plate potentials, that's the potential on the sarcolemma, on an electron micrograph when it was stimulated with different calcium concentrations in the surrounding solution. So this means different amounts of ACH were released with the different calcium concentrations. And they observed that the end plate potential was always a multiple of 0.4 millivolts, and that the end plate potential magnitude was proportional to the number of acetylcholine to nicotinic receptor complexes that were formed. They also recorded the end plate potential with no stimulation and observed these single spontaneous depolarizations of 0.4 millivolts in amplitude. And they called these mini end plate potentials. And this all provided evidence that acetylcholine was being released in discrete quanta or packets of acetylcholine. And this is evidence for the vesicular release of acetylcholine in the cleft. Vesicular release, this theory was clarified when the terminal was washed in fluorescent protein, and you could actually watch that these vesicles were being released and recycled. So the graph here, or from the Fat and Cats experiment, shows the differing stimulus and hence the different change in membrane potential, and shows a spontaneous mini end plate potential, which is one of those vesicles just being released onto the end plate. This is clinical. So myasthenia gravis is an autoimmune condition. Remember we talked about muscle-specific kinase before, which is what helps anchor the nicotinic acetylcholine receptor onto the sarcolemma. And there's autoimmune destruction of the muscle-specific kinase. And hence you have a decreased number of those nicotinic acetylcholine receptors on the sarcolemma in the muscle. So even if there's sufficient ACH being released, there aren't enough receptors. So you get lots of symptoms as listed here: ptosis, diplopia, dysphagia, dysarthria, which gets worse throughout the day. And we can try and treat this with acetylcholinesterase inhibitors. So acetylcholinesterase inhibitors inhibit acetylcholinesterase in the cleft, which allows for an increased concentration of acetylcholine, which allows which allows an increased number of binding to the nicotinic receptors that are left on the membrane. And this is actually often given with atropine. And the reason it's given with atropine, which is quite interesting, atropine is a muscarinic acetylcholine receptor blocker. And it prevents overactivation of muscarinic receptors in the heart, and hence it helps to prevent bradycardia. This is pyridostigmine I'm talking about, an acetylcholinesterase inhibitor. This is another condition. This is not on the spec. This is Lambert-Eaton myasthenic syndrome, sometimes called LEMS. And this is autoimmune destruction of the voltage-gated calcium channels on the presynaptic terminal. So myasthenia affecting the postsynaptic membrane, LEMS affecting the presynaptic terminal. And so incoming action potentials can't cause acetylcholine release because these calcium channels, there's not enough calcium influx to allow that to happen. So you get a weaker muscle contraction, and you have some symptoms which are similar to myasthenia gravis, but it's often less severe. So you can get proximal muscle weakness, autonomic dysfunction, and reduced reflexes. But this is not core. It's just another example of an autoimmune condition that affects the neuromuscular junction. So that's it from me on nerves and NMJ. I hope that was helpful. I'm happy to have any queries sent to my email. I have essay plans from first year that I'm happy to share if people send me an email. Please scan the feedback, because if you give feedback, you can have access to these slides, which I hope are helpful. Thanks for listening. That is everything. I'm going to leave the scanning up for you guys to scan it and get feedback. And please feel free to send me any questions in the chat if you have any now, or if not, send them to my email. And I hope you have a lovely rest of your afternoon, evening in the sunshine.